We are more than glad that Professor Müller could accept our invitation to hold the very first lecture in our series on a topic that is connected to all our lives today. Professor Müller, the floor, or so to speak, the screen is yours. Thank you very much.
I really thank the organizers for the honor of being able to open this series. I'll share my screen in a second. Yes, that's the one. So I hope you are able to see my starting slide here. It was a very hard task to decide what to present today because the field of COVID-19 research is so vast. So it has been in the focus of not just scientific inquiry, but also of public interest. So I expect that all of you are more or less experts in COVID-19 at the moment. So I would like to use this opportunity to put it into a broader context and talk also about one of my favorite topics, which is evolutionary biology. So I'll talk not just about the virus, but about some principles of evolution and how these principles can be used to predict what we can expect from the future of this pandemic. And at the end, I'm also going to talk about the origin, the possible origin of this new infection. So evolution really, as I wrote in my abstract, permeates all life. So it has given rise to the vast diversity of life as we see it today, and we see evidence for it all around us. We see it in the fossil record, we see it in the patterns of diversity among living species, but we rarely see it in action. And there is one field where fast evolution that occurs in front of our eyes is really relevant. And that is the field of infectious disease, of pathogens. So pathogens multiply. They typically multiply fast. They have a much faster generation time than humans or large-bodied animals. They have a high mutation rate, and therefore they can evolve fast. So this is something that occurs on a time scale that can be observed by the human observer. And it has the consequence that infectious disease is in fact not a static entity. So typically we think of established disease as something which is characterized by more or less stable symptoms, stable traits, stable characteristics. But in fact, these infectious diseases are caused by organisms which can evolve, and therefore the diseases themselves can change over time. So why don't we see them all changing all the time? So the reason for that is that most of our infections have been around for quite some time. And there was sufficient evolutionary time that an equilibrium between the evolutionary pressures, between the evolution of the host species and of the pathogen species has been attained. So that's why we don't see fast change, but actually when things change, when circumstances change, then we do see fast evolution. For example, when we are trying to use drugs against infectious disease, then drug resistance can quickly emerge in some cases. And when when you have to do with a novel pathogen, like the new coronavirus in this case, which has had no evolutionary history with the new host species, then this novel pathogen is actually expected to change fast. So seeing evolution now in the novel coronavirus cannot come as a surprise. There are several aspects of evolution, and some of these we can reliably predict, others are not so easy to predict. So one aspect is evolution towards greater transmissibility. This is what we are currently seeing with all the variants of concern that are now spreading all over the world and that are fueling new waves of the pandemic. So this is nothing to be surprised about. So actually, if you think about it, a new pathogen has not yet adapted to the host species. So in the beginning, we cannot expect to be fully adapted, cannot expect to be at the limit of what is possible in the new species. So new variation, new mutations will arise that increases transmissibility. This change is predictable. What we cannot predict is the extent of that change. So that is something that we can only observe. The first observation, this is going to be relevant in this current pandemic, came from early data from 2020. So the first variant which grew to global prominence was a G614G variant. So that appeared quite early somewhere in Europe, and then it grew to prominence. So here in this graph, we can see over time the change in the frequency of this variant. So these codes typically describe some amino acid position in one of the proteins of the virus. So the number denotes the amino acid position, and the letters denote the amino acids that are being changed, so from a D to a G in this case. And this one mutation could be tracked as it spread all across the world. And now this is basically present in all recent infections. Once we have observed such a variant rise to dominance, we can try to find out what provided this advantage, this fitness advantage, this growth advantage to this variant. In this case, we have multiple data now that this variant increases the ability of the virus to bind to the receptor, to the protein that it uses on the surface of the cells to infect the cells. It is more efficient at infecting cells. Now we also have data from animal experiments showing that it is more efficient at being infecting and being transmitted to new animals. So it had a decent growth advantage. In most of Europe, by the time the epidemic grew to high peak, this was already the predominant strain. And then in the recent months, we see an explosion of new variants. If you are a bit confused about the nomenclature of these variants, then you are not alone. So now there are several alternative or parallel systems of how to name these new variants, when to decide that something can be regarded as a new variant. So we have all these codes, but for now, I will just concentrate on the three most concerning variants and use common names like the UK variant for the one that was first identified in England, and then the South Africa variant and the Brazilian variant. Okay, but the number of these variants which are spreading is increasing. Now, we detect these variants because they can grow. So we see an increasing frequency of these variants. And this is the cause for concern. So here we see a global plot of how the main clades, the main strains of the virus changed in frequency over time. And here we see the original variants, the Wuhan strain, so to speak. And then it was being replaced by this mutant that I showed you before. And then within the diversity of that strain, new variants started to emerge. So it's not just a new variant replacing the other. New variants typically emerge from the background of an earlier successful variant. Okay, and this is, for example, the UK variant, which is now that has the highest frequency in the data. These sequencing data are, of course, not completely representative of the global diversity of the epidemic. Some countries do more sequencing, others do less. So this is not a totally representative picture of what is going on globally, but eventually we do see the signature of new variants emerging anywhere on Earth. So just a few variants which are especially concerning, or which received a lot of attention. So the UK variant was identified in the UK, southeast parts of the UK, and in a few months it grew to prominence. So that very rapid increase was evidence of a substantial increased transmissibility. Using mathematical models, the relative advantage of the strain that is spreading on the background of a pre-existing other variant can be quantified. So in this case, it has been estimated that the advantage of this new strain is 40 to 90% compared to the variants that it was out-competing. So that is a considerable advantage. And we have seen in several countries that this can mean the difference between successful control of an epidemic or a new wave of epidemic growing. So these variants are really relevant. Another variant is the South Africa variant. We see it growing also very rapidly in the time frame of a few months. It grew to prominence in South Africa. So the gray area means basically other strains which were not individually tracked in this next strain database. And now it is almost ubiquitous in South Africa. And then these variants, of course, do not respect national borders. So all of these variants are spreading all across the world at the moment. Then there is the Brazilian variant, which appeared in Brazil. And it also grew to prominence in a few months. And it fueled a very severe second wave of the epidemic, a second wave of deaths in Brazil. The reproduction number, which is basically a single number characterizing how transmissible pathogen is in a population, was estimated to be increased by 70 to 140%. So this is also a huge increase. I'll come back to this later. So this increase can actually include two things. One is a generally higher efficiency of infecting cells or being transmitted. And in countries where we already have widespread immunity against previous strains, like Brazil very likely had it before the second wave, then evolution that allows the virus to escape immunity can also provide an advantage. So probably in this context, both changes played a role in the success of this variant. So this relative increase is valid with respect to the population of Brazil. Then we try to map what makes these variants more efficient, and we can find some common mutations, like the one I indicate here has now been shown to increase again the binding efficiency of the virus to the molecule that allows it to infect cells. We also see that this 614 mutation is now in all the variants that are currently spreading. And then there are further mutations. To some of these I'll come back later in a different context. And what we see in most of the countries is now a mix of variants that are spreading and competing with each other. And soon we will come to a war of variants when more than one of these efficient variants will arrive into the same countries. And this is sometimes already observed in some of the countries which have a more international travel. I showed two examples where we have relatively large number of sequences, so where we can make reliable inference. And so here in Luxembourg, what we see is that this is the UK variant, and now we have an increase of the South African variant in the background, on the background of the UK variant. In Belgium, we have a recent increase of the Brazilian strain. So these things are quite concerning. If these variants now, having met the UK variant, are able to grow even on the background of this highly efficient variant, that is not good news. So we can say that indeed we could predict evolution towards greater transmissibility. And this is occurring, and unfortunately the degree of this process is also substantial. Now, a very important question is not just transmissibility, but virulence. So when you are infected, what are your chances of being severely ill or dying? So virulence in general is the harm that the pathogen causes to the host. So this is also a very important aspect of the evolution of pathogens. And here I would like to give you a more general background on the evolution of virulence, what to expect from what we know from the example of previous pathogens and diseases. So in general, evolution by natural selection occurs if there's heritable variation in some traits that affect reproduction or fertility. And in the case of a pathogen, there needs to be traits that affect the spread of the pathogen and that have heritable variation. Now, virulence in particular is going to evolve if heritable traits that affect transmission are also linked to virulence. But this is almost trivially fulfilled. So a lot of traits that affect the life cycle of the pathogen will affect how serious an infection is going to cause. If any trait increases the efficiency of the pathogen, it will replicate to a higher level. Whatever harm it causes to the host, it will cause more harm. There are also side effects by which the pathogen can cause harm in many cases. So these can also evolve independently. Basically, almost all of the traits of the pathogen could affect virulence, and many of them also affect spread. So a link between some of these is almost inevitable. That means that basically the severity or virulence of infectious diseases can evolve, but it's very hard to predict in which direction. There was the traditional view, and many people still hold this view, that given sufficient time, host-pathogen interactions evolve towards benign coexistence. There is a logical explanation for this view, saying that if the host can survive longer with the infection, then it can transmit the infection longer, so that facilitates spread. And this is a corollary that those infections which are still virulent, these must be evolutionary recent. So the new host species was colonized recently, and we do see very conspicuous examples for that, like Ebola virus, or the Legionella bacterium, or the SARS coronavirus. The original SARS coronavirus in 2002 was also that belongs to this category. So these pathogens are not continuously present in the human population. There has been no coevolutionary past with humans or between humans and these pathogens. And we see that when they do infect humans, we have very high mortality. So Ebola virus, in some outbreaks, it could have mortality going up to 90%. But even in the largest epidemic, 2014, there was somewhat reduced mortality or fatality rate, but it was still 40%. The original SARS coronavirus also had a mortality of 10 to 20%, and the Legionella bacterium can also cause high mortality. But if we look closer, then there are actually also counterexamples. There are infections which are ancient, so which have had plenty of coevolutionary time with their hosts, and yet they remained virulent. malaria is one of the greatest infectious killers, and it has been so for many, many thousands of years in human history. Now we know that the most aggressive variant falciparum malaria entered from gorillas, and it doesn't show signs of losing its virulence. Measles, before vaccinations enabled us to control it, caused a very high level of mortality in children, either directly or indirectly, as we now know it. It's estimated to have entered the human population, the human species, about 1,000 years ago from cattle. From a virus of cattle was the ancestor. And then smallpox was one of the most virulent diseases in human history, which persisted for thousands of years. So it probably originated from some sort of rodents which have similar viruses. And again, it showed no signs of evolving towards benign coexistence. So that is not always happening. This figure actually illustrates the logic behind that. So now what we call the trade-off, this is the trade-off principle, which governs the evolution of many traits. It means that one trait can only be improved at the expense of another. And in this case, the trade-off is linked to different consequences of how fast, let's say, the organism is replicating. So if the pathogen replicates faster, then infectiousness in the host increases. So you had a higher level of the virus, more virus in an individual. That's good for the pathogen. So you would expect there to be selection pressure towards faster replication. But on the other hand, whatever negative consequences the presence of that pathogen has in the host, if there's faster replication, these consequences are going to be more severe. There's more exploitation of the resources of the host. A stronger immune response is invoked. So in any case, the length of the infectious period is going to be shorter. Either the host can die faster, or it will launch a stronger immune response, and that reduces the length of the infectious period. So that has a cost for the pathogen, and these two factors actually go in opposite directions. So that says there is no general principle that would say that faster replication is always better, or slower replication is always better, which would mean that you spare your host. It is not necessarily better because you can lose in infectiousness. If you have such a situation, there is not a single solution, but what happens where the optimal rate of replication and therefore also optimal virulence for the pathogen is, that depends on biological details of the system, on the shape of these links between replication and length of infectious period, and replication and infectiousness. So this cannot be predicted in general. And in practice, this is going to be different for different host pathogen pairs. Okay, what we can derive in general, and I won't go into the details, but this is a general principle that given these opposing factors, there will be an optimal level of virulence for each pathogen, host pathogen pair. The evolution of the host also matters somewhat. Of course, there is a response from the host, but the evolution of the pathogen typically dominates this arms race because it can be faster, but eventually can settle to an equilibrium. So where this equilibrium is, or where the optimum for the pathogen is, depends on a lot of details. But without knowing these details, just think in terms of the whole spectrum that you have from herpes viruses, which are basically harmless in humans, almost always, and that goes up to smallpox. And in animals, we know infections that are even more fatal, have a high fatality ratio, which does not decrease. Now, given this, now we know that virulence does not always go to a kind of negligible level. There is an optimum. So with this knowledge, now we can expect, or we can infer, that when a novel pathogen enters a new host species, we have no way of knowing whether it is below or above the optimum in that new host species. The initial level of virulence is just a matter of chance. There was no prior coevolution. So it can be either below or above, which means that after the host jump in the new host species, evolution can drive the pathogen and the infection either towards higher or towards lower levels of virulence. Both can occur, and we have seen examples of that. So that means there's no way of predicting this in general. Now, what do we see in the case of the new coronavirus? What we see is that the UK variant is probably considerably more lethal than the original variant. So there are some studies which still cast some doubt on this, but I think this result is quite reliable. So that is bad news. That is bad news, of course, in the countries where the UK variant is spreading, but this is also bad news in general, because the rules that govern the evolution of this new coronavirus are likely to be the same for the UK variant as for the other variants. So that means that in this case, probably there is some linkage which makes some traits beneficial for the virus, which also increase virulence. So that means that similar parallel trajectories of virulence evolution can be expected also in independent lineages, in independent variants of the virus. And we don't know yet if this is the case, but perhaps the high mortality now observable in Brazil and India could also partly be due to an increased virulence of those variants. The next question is whether we can expect or predict evolution towards immune escape. Immune escape is the process in which the pathogen changes in a way such that it becomes basically resistant to the effect, or partially resistant to the effect, of an immune response which developed in a host that was infected by an earlier variant. That is immune escape. So that can lose immunity, the protective effect of immunity. When do we expect such an escape or evolution towards immune escape to occur? When there is fast initial spread of a new pathogen in the population, like it occurred in the first wave or even in the second wave in some countries, then most of these infections occur in a immunologically naive population with no prior immunity. And what we have in most individuals is an acute infection, an infection that resolves in a few weeks' time in almost all of the cases. This is such a short time that actually the immune response that is developing has an effect on the virus only at the very end of this period. So there is a short window of time when there is selection pressure on the pathogen to evolve escape from immunity. And actually, this is what we see. So there's no strong evolution typically seen in the first fast waves of pandemic spread. Then there are specific situations in some patients who have, let's say, immune impairment, some form of immune deficiency. One could have long, almost chronic infection that lasted for months. And these patients were treated with drugs. They also launched some partial immune responses in some cases. This is a different selection scenario. So here you have the immune response kicking in, and then the virus still persisted for weeks afterwards and was under selection pressure to evolve immunity. So when these new variants occurred, and actually some of these most concerning variants occurred in a way, in a surprising context. So these were these represent relatively large jumps in evolution, as opposed to the slow, continuous accumulation of changes. And some people, some scientists suspect that these variants might actually have evolved in chronically infected patients, because there you have strong selection pressure to adapt to that individual. And now we have actually detailed studies from such patients monitoring the evolution of the virus, and we see this is happening there. Then after some time, eventually we end up with a partially immune population. In some countries where there were no strong lockdown measures, the infection wave itself immunized large parts of the population, or vaccination can also immune large parts of the population. Then the selection regime changes again. So now it can occur that the virus is moving from immune or partially immune individual to another one, and is under constant selection pressure to be able to avoid that immunity. If immunity is perfect, that of course stops this. So then if someone cannot be infected, then there's no selection pressure. Partial immunity is the problem, but immunity declines over time. So sooner or later, everyone arrives at that stage with partial immunity when reinfection can occur. But there is still some partial immunity which imposes selection pressure towards immune escape. So that is a dangerous situation. And the country where we saw it best is Brazil. So in Brazil, many parts of Brazil, the first wave was so huge that it was estimated to immunize the majority of the human population. So people expected a long break from further waves, and it was actually a tragic surprise that less than half a year later, an even stronger second wave started, and a deadly one. And then it turned out that this was fueled by this new variant, by the Brazilian variant. This is P1. And it turned out that it not only has greater transmissibility, but protection from previous infection with the original variants, with non-P1 variant. That protection is greatly reduced against this new variant. So that, again, is not good news. And now we have some idea which mutations are responsible for this. And these mutations, unfortunately, appear also elsewhere. When the immune system encounters a new pathogen, then there is variation in what the immune system can target. But there are some conspicuous targets which are often targeted by the immune response. And in this case, this is also this spike protein which enables the virus to attach to cells. This is on the surface, this is visible, and binding to that is actually what gives protection. I mean, binding of the antibodies, what gives protection against infection. So it is not surprising that changes in the spike protein can reduce the effect of previous immunity. And there are several of these recurring mutations are involved or seem to be involved in an escape from previous immunity. So what does this tell us? So one message is to those who have just received their first vaccine shot. This is a time when you have to be extra careful. I mean, this is a time when you are already impatient, you would like to go out. You know that your protection is now building up. But think about it. So this is a stage when you are developing partial immunity. After a few weeks after your first shot, you are already pretty well protected from serious infection, but you can still acquire it and you can pass it on. And if you are at that stage, then you are the perfect breeding ground for immune escape for the virus, because now it can still replicate, but it is already under selection pressure from this partial immunity. So for the sake of your fellow human beings, I ask you to be extra careful and be a bit more patient in the time after you have received your first vaccine shot, but you are not fully vaccinated, fully immune yet. Okay, so obviously these mutations can affect vaccine efficacy or efficiency. We have a lot of data on that. There is some variation as to which variant abolishes or how much they reduce protection offered by the different vaccines. What we see that partial protection often remains. And what is a hopeful sign, so I've had some bad news so far, but a good news is that the vaccines can be updated to include these new strains, and they are being updated already. There are also data showing that some of the vaccines actually provide better protection against these variants than exposure or infection with the original variant, or that boosting immunity by from an earlier infection with the vaccine can improve your immunity also against the variants. And a further bit of good news that I can offer is that when you have had multiple exposure against several variants, let's say, and you probably survived that because you are vaccinated, you had partial protection, and then you are also immunized again with a variant, then your immunity becomes broader. So immunity is not an isolated phenomenon that you are immune to one variant and then independently to another. Immunity is like a cognitive system. It learns. So when you have been exposed to several variants, you will build a broad immunity, which will also protect against further variants. Immune escape has limits. So it has limits what changes the virus can still tolerate and remain efficient. So it cannot escape all the time. Okay, so we can predict that under the current circumstances now, immune escape evolves because more and more people are partially immune. Now, what about drug resistance? So we have some drugs that have some effect against the virus. We are not totally happy with them yet because we don't have a cheap and efficient drug. But several drug candidates are in the making. Do we expect evolution towards drug resistance? That's an important question. With several bacterial infections, drug resistance is a huge problem, and we know that it can also evolve very fast in some viral infections. Now, this is a figure that shows of one of the possible drugs or drug actions. The virus produces several of its proteins in the form of a polypeptide, and then it has to cut them up, and this is done by an enzyme of the virus. And if we develop a drug molecule which can bind to this enzyme, blocks its action, then the virus cannot infect new cells. It cannot, won't have properly functioning proteins. So that's a very promising avenue for drug development. But of course, if the protease changes in a way that it no longer binds to the inhibitor, then this effect is lost. Then drug resistance emerges. For example, HIV, the human immunodeficiency virus, that can evolve resistance against protease inhibitors. So what do we expect here? Okay, in chronic infections, like we said, there's a long time window when the virus is exposed to the drug, then it can evolve resistance easily and quickly. But here we have to do with an acute infection. So almost always this is an acute infection, and that means that the infection is resolved. It's a short period of time. So if we have an active drug, I don't expect a strong, fast evolution towards drug resistance, which is good news. Okay. So it might occur, but I don't expect to be prevalent. Now, what about the long-term outlook? Okay, we don't know yet how far this is going to go. We have seen that evolution towards increased transmissibility is in a way inevitable, but it has an upper limit. We don't know how close we are to that yet. So it might still continue evolving towards higher transmissibility. We don't know that. Okay, there is one thing: if we have partial immunity, then actually optimal virulence can increase, and that's a rare but a possible phenomenon. But we have seen, or I've explained, that immune escape can only go so far. It cannot go on forever. So eventually, I expect population immunity, and I'm not alone with that, to build up and basically stop this virus from becoming virulent, from being virulent. Evolution of the host can also occur, but this is not what we want. The evolution of the host in this case would mean that those humans who are particularly susceptible due to inherited factors, they die fast, and then our species evolves some form of resistance to the virus. This is not what we want. We want to avoid the infection by not acquiring it, or by vaccines, or by drugs. Now, I also call attention to the fact that the epidemic can change substantially without the evolution of the virus. For example, many people believe that it will transition to an endemic mild infection. We see that it is mostly aggressive in elderly people, chronically infected, people with chronic conditions, health conditions, and not in young people. That's the general rule. There are exceptions, of course. And once the virus or vaccination has immunized the majority of the population, the remaining susceptible individuals are going to be the young children, the newborn, who have not been exposed yet. But then, even now, they would be less prone to serious infection. So if nothing would happen, even then, we would expect a gradual change towards reduced death toll. But we can do better. We can speed it up with vaccinations. So that much about what has been going on and about the future of the virus. And I would like to give you a few words on the possible origin of the virus. I see that time has already flown by, but let me try to do this in, say, 10 minutes or so. When we realize we have to do with a new pathogen, what do? So first we'll look at comparisons. What does it look like? What does it resemble? Even in a micrograph, we can see that it is a coronavirus. It resembles coronaviruses that we have known, but we don't have to rely on our eyes or things like that. We can rely on the genetic information of the virus. So this is a series of a string of amino acid letters that we see in the different variants of the virus, and we can compare these. And evolution leaves traces, it leaves fingerprints in the genome, so we can use this not only to decide who is related to whom, but also to infer the time of when two lineages branched from each other. What could be the possible origin of a new virus? It might occur by evolution of another human virus that has changed, that has acquired some new properties. That's in principle possible. Or it could arise from an animal virus. I'll come back to this, that this has happened in evolutionary history. Or there are many people who have some suspicion that it might be an artificial construct of some sort. So I will also talk about that possibility. Okay. When we construct the phylogeny, which is basically the family tree of related viruses, then we see this. So here, these are the names of different viruses. Those that are linked to each other are closer relatives. And then we move back in time. It's like the generation of your grandfathers and great-grandparents and so on and so forth, move back in time. And what we see, these with the yellow letters, yellow font, these are the four coronaviruses that cause common cold, and they have been around in the human populations for quite some time. Then there's the MERS coronavirus and SARS coronavirus, which infect or infected a small number of people, but with high virulence. And now we have the SARS-CoV-2, the virus that causes current pandemic. And what we see immediately is that these human viruses have their root, they join to this tree of a sea of non-human viruses. They have independent origins. So the common ancestor of the two SARS viruses was not a human virus. It was an animal virus. It is in the background of a diversity of animal viruses. This is basically true for all of these. So we have independent origins for these viruses, which means it must mean that it came from an animal. So we can exclude the origin from a human virus. Now, could this be an artificial construct? So the narrow sense of an artificial construct would be that someone used some sort of genetic engineering to create a virus, which was not natural, not present. This is certainly not the case. So all the techniques that we could use for such artificial manipulation of the genome, they leave traces. There are no traces of this in the genome of the new coronavirus. So it is definitely not an artificial construct. It doesn't entirely rule out the possibility of human action or responsibility. I'll come back to that later, but it is not an artificial construct. So where could it come from? We know that many of the important infections of humans came from animals, actually. So the ancestral pathogens can be found in animals. In this case, the closest related viruses are typically in bats. Bats harbor the largest variation of the viruses that are related to the new coronavirus. But the pangolin, this strange mammal, was also implicated. I'll show why. So what we can do is compare the genomes, the genetic material of these species. And here we see on the horizontal axis the length of the genome, and on the vertical axis we see similarity to the new virus. So the different lines represent different animal viruses, different animal coronaviruses. And here what we see that the closest related species, this black line, is a bat virus. Since then, this has something like 96% identity or similarity to the new human virus. Since then, another bat virus was found, which is a bit even more similar. But there's one region where the similarity drops substantially. And this is actually the region which is responsible for binding to the surface of the cells. Okay, so this is the protein, this codes the protein which binds to the receptor protein on the surface of cells. So this is actually what determines what kinds of cells can be infected. So a divergence there is not at all surprising. So this is where you would expect the greatest difference between viruses that infect different species. But the thing is, most of these bat viruses cannot or can only inefficiently infect human cells. And that's where the pangolins, the strange mammals, come into the picture. The general similarity of the viruses is lower than that of these bat viruses. But in this region, and here what we see on the bottom graph, that's a magnified picture of this region, so the critical region. Here we see that the pangolin viruses, this purple and the green line, they have closer similarity to the human virus than the bat viruses, and there are actually 6 positions in this whole sequence of amino acids which are critical for the binding, and in those 6 positions there is a pangolin virus which is identical to the human virus. So how could such a situation arise? It could arise if the human virus came from a background of the diversity of bat viruses, But there was rapid adaptation of the binding site to a new host. And this might have occurred in humans, but we don't see those early infections. We have no evidence for that. What we saw was already efficient spread in Wuhan, in the epicenter of the epidemic. All this rapid adaptation could have occurred in an intermediate animal species. So perhaps there was a jump from a bat to another mammalian species, which had a receptor on the surface of the cells that was closer to that of humans than what we see in bats. That's a possible explanation. Or recombination can do this trick. So instead of gradual evolution, coronaviruses can mix their genetic material. So two different viruses, if they manage to infect the same cell, then they can mix their genetic material, and then we can also, such patterns can arise also by that. We don't know which explanation is correct, and we don't know where and when this might have happened. When we do this phylogenetic tree and we infer the times for the splits, for the branch where the branches split, then what we see that the split between the novel coronavirus and the closest bat virus was more than 40 years ago. So that's a long time. And what we see is that all the variation of the current virus, it goes back to just the end of 2019. So that's where the epidemic started. And that means there are 40 years of evolution unaccounted for. That means we haven't found yet the immediate source of this infection. There must be an animal virus, which is very close to the root of the human pandemic, to the root of the viruses of the human pandemic, and we haven't found it yet. Now, the where is also an interesting question, because the closest bat viruses live in Yunnan province, and the epidemic started in Wuhan, which is here. So that's quite a bit of a distance. But then, of course, we also don't know if the original species was in Yunnan. We have no way of knowing that. What we know is that this is the closest one, but that is still removed by 40 years of evolution. Okay. And then, of course, this is the part of the question which interests people the most. Was it a direct jump from animal to human? Or there's an alternative theory which says there was a lab leak. There might have been an accidental release of a virus from a lab. And before I go into the details, let me say at the moment there is no direct evidence for either of these theories. We just don't know. Okay. I'm still saying that this is not the field of conspiracy theories. This is a valid scientific discussion because it has consequences for the future, and also going whether we are ever going to be able to find out. And I'm close to the end of the talk. Before I go directly into what we know so far about this, let me do a brief detour to explain what a gain-of-function research is. A gain-of-function research is where you do experimental evolution and manipulation of microorganisms so that they acquire new functions. Okay, and one example for this, when you evolve or manipulate viruses or other pathogens to change their host range, so what species they can infect. Now, who would alter a dangerous pathogen towards the ability to infect humans? You might think that this is out of the question. No one would ever do that. But this is unfortunately not true. A historical precedent, just less than 10 years ago, there were some experiments which changed the bird flu strain that was circulating in birds and very aggressive, virulent in birds at that time, H1N1. So this is influenza. And there were three independent experiments, one from the Netherlands, another from the US, but from a Japanese group, and the third one from a Chinese group. And they did just that. So they changed deliberately the bird flu strain to be able to infect mammals. So moving much, much closer to to being able to infect humans. And they did that basically in a good intention. They did that to be able to map the risk, to map the possible evolutionary routes of this virus adapting to humans. But there was a total public outcry and an angry reaction also from many of the experts, with whom I totally agree. The reason for that is that we have no way of knowing if these experiments are ever going to be useful. There are alternative evolutionary trajectories. There is, I think, and many people believe that, very little risk that we actually find a useful outcome from these experiments, and that will help us to fight an eventual pandemic. But we greatly increase the risk of such a pandemic occurring because we create viruses which otherwise might not have evolved. So this strain of bird flu, for example, in the last 10 years showed no signs of evolving to become able to infect humans or other mammals, but they did it in the lab. And we also know that there are lab accidents. Even in the best guarded cases in the US, in the UK, in the Europe, there have been accidents where pathogens can be released from a lab. So this is really playing with fire. And the nasty human side of that is that the three experiments got the authors' papers in the most respected scientific journals of the world, Nature and Science. So of course they are motivated to do that. So there was a public outcry, and then there was a temporary moratorium, at least in the US. Several other countries also joined, not to do such experiments. But not all countries joined, and the moratorium has since been abolished also in the US. So such experiments are being done with greater scrutiny and all caution, but they are being done. I think they shouldn't be. And so that's why it's kind of a legal debate to talk about this as a possible origin of this virus, because probably didn't happen, but it could have happened. So such experiments have been done with other viruses. And the bottom line is they shouldn't be done. Okay, it could have happened. Even if it didn't, this is a lesson that we should be more careful. And let me totally stress this. There is absolutely no evidence it did happen, okay? I'm just saying all this to show that this is a dangerous path and we shouldn't be doing it. It could have happened. The final part of the argument is that it did not need to happen. This is not the only way such a pandemic could start. And actually there is a simpler explanation, a natural explanation for this, how this might have started. And here I put together some of these arguments for and against the theory. Okay, so some people find it suspicious that we have the Wuhan Institute of Virology right at the epicenter of the epidemic, and the closest form of the virus is far away from Yunnan. Okay, that's true. But we know that that virus is actually not the direct source. So we have an unknown source, and these bats are living all around China, so they could be anywhere. And there are live animal markets also in Wuhan. So there is possibility of contact with infected bats or other animals and humans. So the natural transmission might easily have occurred, or perhaps it occurred elsewhere and someone just traveled to Wuhan. We know that this infection has a long latency period. An argument is that it's too well adapted to humans. So already at the start, it was pretty much able to spread. That's true. That's special. But these viruses are special in that respect. So now we see that this group of viruses, from which also the new coronavirus comes, they are particularly flexible in their host choice. So they have switched between hosts more often than is typical for other infections. And also we know why that can be the case. These receptors that they use to enter the cells, they are very, very much conserved, as we say it in evolution, which means they're almost the same in many mammalian species. So that's why they have an easier time switching species. So this doesn't need specific artificial explanation either. Finally, we can say that, okay, so now there is focused research on SARS like coronaviruses. Okay, so maybe the risk was higher, but think about it. Before the first SARS epidemic started in 2002, we had no idea these viruses were dangerous. They were regarded to be totally harmless for humans, and hardly anyone worked with them. So the first variant in 2002 certainly emerged without kind of human help. And that also means it might have occurred again. Okay, so this is all the evidence, and this is basically lack of evidence that we can put together in this case. Are we going to find out? The only way to find this out to a certain direct animal to human transmission would be to find the virus which is very close to the human virus. An animal virus which is close to that has the same kind of receptor binding domain, and we find the epidemiological link to this population. This might or might not occur in the future. Until then, the bottom line is there's no basis for accusations at the moment, no evidence. There is need for further investigation. And I would like to add, as a moral of the story, whatever happened now, do no gain of function experiments that enable pathogen for human transmission. And now this is the summary slide. So you see that the origins are still a bit veiled. We know the ultimate origin comes from a family of bat viruses. We don't know the immediate origin yet. The present is bleak. At the moment, evolution helps the virus to evolve more efficient variants, or those that can evade the immune response. But the future is hopeful. Even this pathogen evolution has limits, and our immunity evolves too. So ultimately, we are going to end up with broad population immunity due to vaccines, I hope. And the bottom line again, so I wanted to present this coronavirus story in the frame of evolutionary theory to show you the power of evolution that affects even our everyday life. And I thank you for your attention, and I stop sharing my screen, or at least I try to.
Thank you very much, Professor Müller, for this very, very informative and very interesting lecture. And I would like to inform all the participants, and actually call on all the participants to raise questions in the chat, which then I can put to the screen. There you see the first one. How likely is that such a pandemic happens in the near future, or can it be predicted how frequently pandemics would break out in general? And I will also have a kind of a similar question. I would like to ask the professor to answer this one.
Okay, so these events are so rare that we cannot use statistics to kind of estimate the frequency. In the historical past, huge epidemics like the plague epidemics occurred every 600 to 800 years. So sometimes shorter, but with large gaps in between. What we can say is that now we have a world which is more interconnected than it has ever been. So that certainly increases the risk of some kinds of pathogens spreading in the human population. And I think it's not just a single instance, it's probably a general rule that respiratory infections have a better chance of spreading. Many infections that were linked to hygiene conditions are unable to spread at the moment. So those that are spread by the oral fecal route, for example, like cholera, whatever, most parts of the world are unable to spread. Neither are those that required, like plague, which was the greatest killer that requires transmission by rats. So these things have been basically dealt with by technology. But with respiratory infections, we have a world which is more permissive for these epidemics than it has ever been. So we can probably say that the chances are now higher than they used to be. But I think there is no decent scientist who would say that, okay, then it will happen every 10 years or every 15 or so. We just don't know yet.
Thank you very much. My question would, if you allow, I connect my question here because that is, of course, I'm absolutely out of the field. Actually, I come from the field of philosophy, but I found one point which is interestingly connected. I heard you use many times the expression that something cannot be predicted, but you never use the word accidental or accident and any related terms. So this means that when we say, and it is really just a theoretical question on your field, when you say something cannot be predicted, this doesn't mean that these things would be accidental, right? It just means that there are so many components that it is just simply not possible to calculate with them mathematically or statistically or in any way to tell something in advance about what would happen. But when it happens, looking back, we won't say it was accidental. Do I see this correctly, or is absolutely not the right approach?
Well, I think we use accident in the sense when there is unknown variation in the conditions that cause something, and that is always the case in natural science. So we never have perfect knowledge of the system. For example, when I say the direction of the evolution of virulence cannot be predicted for a new pathogen, that is because we don't know yet all those intricate rules that govern this evolution. When we have already studied an infection closely, then we might be able, or sometimes we are able, to recognize these rules, and then we can actually predict optimal virulence. So such things have been calculated for HIV, the human immunodeficiency virus, for example. But with a novel pathogen, we don't have that knowledge yet. So that's why I'm saying we are not able to predict it yet. Also, when we think in terms of new pandemics, so that is the realm that we're never going to know, I think. So many details will influence the probabilities. So a very rare event can happen somewhere that the news, then let's say a hunter comes in contact with an animal somewhere, and then whether the pathogen of the animal just had, by bad luck, a mutation which was not terribly useful in that animal, but now it enables the jump to the human. So normally the animal virus is not able to jump to humans, but it generates mutations all the time. So one way these jumps can occur, that such variants which are kind of pre-adapted to humans can occur, they disappear. And normally you don't even ever see them because we don't have the bad luck of a human stumbling upon that particular animal. But very rarely that happens. And then that rare event can have a huge impact afterwards if it grows into the pandemic. Or think in terms of chance. I mean, also this pandemic started with one infected individual, ultimately, at the very beginning. Had that infected individual isolated himself or herself in the beginning, let's say in the forest or whatever, just never met anyone, then the pandemic would not have happened. So at the very beginning, there is always a large role for chance. If this is a pathogen which is efficient at spreading, then after you have had a handful of cases, then it's out of control. Then chance plays a much smaller role.
Thank you very much. We have another question also related to, how can we know if a genome is artificially created? Are there any specific parts which suggest this?
So artificially created would mean that we basically insert part of another genome or a synthesized piece of genome into the original. But we don't have really scissors to do that. We have molecular scissors, but these systems have pieces which are themselves integrated into the genome in this process. So that's how we would see if that was the origin of a new pathogen, and we don't see that. One thing I wanted to add that on the chance of a new pandemic. So even if we cannot predict it completely, we can still take some steps to reduce this unknown probability. So in this case, now we know that there is a group of viruses which poses a threat. They are mostly present in bats in China, right? So then one precaution is to reduce contact between these bats and maybe also between other wild animals and humans. So let's shut down these live animal markets. And then we have probably blocked the most important entry route, most important entry point of these viruses.
Thank you very much. The next question says, what would be, and again about projection, the projected timeline for vanishing of this COVID-19, if it is possible with the help of mathematics simulation perspective?
Okay, so vanishing, this is not so much a mathematical question. There have been modeling efforts to kind of map the possible scenarios. But again, the unknown factors are so numerous that you cannot use modeling to pinpoint which is going to happen. You can try to quantitate or semi-quantitate possible trajectories. And this is what modelers have done in this case. So in this case, modeling is more useful for short-term predictions than for long-term predictions. But we can rather use our virological and immunological knowledge and our comparative knowledge of early infections and thinking what would happen. So I think the consensus, what most scientists believe, is that eventually, and I think not so far from now, this is going to transit to a mild infection. So as I said, once you have efficient immunity, then it might not be what we call sterilizing immunity, completely blocking infection, but it will greatly reduce the chance of severe infection, severe symptoms. My intuition is, and not just mine, of course, that if you were someone living in an isolated Pacific island, and then for the first time you meet someone from other parts of the world, and you acquire a totally harmful looking common cold virus, then it could also cause severe infection in someone who has never been exposed to those viruses. So we are exposed to those all the time. We acquire them. We first contract them in our childhood, when the immune system is not so jumpy yet and more flexible at learning new pathogens. So then we get through this mild infection, and by the time we grow old, we already have accumulated very efficient immunity against those. So this is what I expect with the new coronavirus as well. So the first waves are lethal because now adults and also elderly people are exposed to it for the first time. And it's very different from all the other viruses that we have met before. So our immune system is not able to respond to them efficiently. But even in the elderly, we see that efficient immunity can develop. Vaccination in particular can induce efficient immunity. So once we reach that, then I think even if this virus doesn't go away, very few people believe it will go away now. It will change into a harmless infection.
Thank you very much. And then the last question we have asks, do you think that this pandemic has affected the scientific work world? If yes, how?
I think it has accelerated some processes which have been happening, but now it just kind of jumpstart this. So we all had this online distant meetings, for example. I think these will also stay in the scientific world, less conferencing and flying here and there. I hope it will remain because this is just so efficient and environmentally friendly. Also, the sharing of data that was boosted. So this is also a trend which has been going on for quite some time now, but in terms of the coronavirus research, it really very fast reached basically a total consensus in the world that these data have to be shared. So this research was, and has been, still is, so urgent. We don't want to wait for the long process of checking the publication, publishing it, many months of delay. Much of these results were just put to preprint servers. These are before peer review. I just show the world what I have done. So I think this process has also reached greater acceptance in the scientific community. I think it's going to stay also after the pandemic.
Thank you very much. And if we have, meanwhile in the chat, we also have a gratitude, a great thank you from an attendee. And if there are no more questions, then I would like to thank again Professor Müller for the fascinating lecture, and of course all our participants for joining the first lecture of the What's New in Science series, and the very first event with that of the ELTE Alumni Academy. If you are an alumna or alumnus of ELTE, please don't forget to join the university's alumni community at alumni.elte.hu, where you can read interesting articles, stay informed about the events, find different opportunities, and join the groups, and connect with the ELTE alumni network. So always remember, your ELTE alma mater welcomes you back. Thank you very much again for participating, and thank you again, Professor, for the great lecture today. And everyone, please stay safe and healthy.
Thank you.